controlling viral replication. But when you see it persist past this peak of the virus, that's when you might run into complications in terms of resolution of the infection and repair processes. And we see this sort of niche window where you downregulate your interferon responses and you see this onset of epithelial repair. And so our model sort of tries to dissect the two and see if there's an interplay.
Yeah. So, you know, and your lab, Andreas' lab, has like studied interferons in viral infections since a long time, and you know, giving interferons early always worked really good. Interferon lambda is really beneficial. I mean, it's something that is known since a long time, right? Is that correct?
Yeah.
But interferons, nobody disputes that interferons are antiviral cytokines and are very efficient into viral infection. You know.
To expand on this also, I mean, prior to the COVID work, my projects were on interferon modulation and non-small cell lung cancer, and we see a similar thing where early interferon responses really bias IgG signature and immune cells, which is a positive thing. But then when you have it long term, you see the IgG enrichment in the cancer cells, and that obviously becomes more likely of a relapse. So it's a similar phenomena. It holds true in multiple different settings.
All right. That's interesting.
Sounds like we'll have a lot of discussions to do. Yeah, but I think the phases and having longitudinal data will be very key and very interesting to see as more and more comes by, for sure. I guess let's take another 2 minutes to wait for others. And for everyone who's already joined, you can see a lot of things on the chat box. And also we're actively looking for people to co-host and organize symposiums. So please stay tuned. Please make sure everyone joined so far has their mic muted. But if you're not a speaker, please make sure that your mic is muted. And as you see on the right, you can type in your questions if they arise anytime throughout the talk. And Nicholas will help ask the speakers either at the end of a talk, if there's time, or towards the end of all sessions. We'll give it another minute for everyone else to come in. Sorry, there's. Okay, let's get started. So everyone, welcome to the Open Box Science Talk. Today, this is a little earlier than usual. So thanks for joining early. If you're in the US time, I think we're trying to be accommodating for people who are in Europe and Asia. And it's really tough when we have more global audience to find the time that works best for everyone. So thanks for showing up. And before we start, as you probably all know, Open Box Science is this effort to make Journal Club available everywhere to everyone around the world so we can all come together and discuss our scientific idea. And in these seminars, we hope that obviously we all satisfy our drive to understand and curiosity to understand science better firsthand from first authors. So we are very lucky to have 3 first authors, as you will hear more today, presenting to us today about their exciting papers surrounded COVID-related immunology. And I think it really is an extra experience if you host a symposium because you get to get in contact with the authors. You can write in your CV of hosting large-scale talks, which only happen really late stage in your career. But it really is always better to get more practice giving talks and hosting talks early on. And then finally, you can really get to know other scientists, especially when you're at a stage where you're potentially looking for a job or looking for young talents in your lab. So with that today, our seminar today is co-hosted by Nicholas, who's assistant professor at Mount Sinai, working closely with Nina Bartwatch in the immunology department here, or Immunology Institute at Mount Sinai. And he really also is an expert in tumor immunology, as well as recently, I think he's been leading the pretty famous Mount Sinai Immunology Review effort, which has posted hundreds of critical reviews of COVID-19 related preprints. As we know, there's a critical amount of them, and gather and compile these evidence for the field. So today I will let Nicholas moderate the talk, and we're always happy to answer on the side, on the chat. If you guys have direct questions or technical questions, you can directly just chat privately to me. Throughout the talk, please make sure you're muted, and you can type your questions if they arise, and we'll try to make sure we have time to address them afterwards. So with that, I'll let Nicholas introduce Jack and start today's symposium. Thank you.
Thanks, Quan. So welcome everybody to this symposium on COVID-19 immunology. So our 2 first speakers today, we published 2 back-to-back papers in Science showing that the interferon response, and in particular the type 1 and type 3 interferon response, is harmful for lung barrier integrity after viral infection. And I think it could not be more timely because it's becoming more and more clear that the interferon pathway is central to COVID-19 pathophysiology. And now we have data that SARS-CoV-2 is particularly efficient at avoiding the induction of interferon response. And we also have data from patients showing that there is a profound dysregulation of the interferon pathway in immune cells like myeloid cells. And I think this point will also be detailed by our third speaker today. And I think maybe what's the most striking to highlight the complexity of the role of interferon in COVID-19 pathology is the fact that there are 2 different therapeutical approaches which are seemingly opposite. We have seemingly opposite effect, but actually work in patients. So the first one is the administration of recombinant type 1 interferon. And the second one is the inhibition of the JAK-STAT pathway, so JAK-STAT inhibitor. And both of these therapeutic approaches seems to show promising results for the treatment of patients. And there's probably a lot of more mechanistical understanding to get to exactly understand what patient would benefit the best from actually treating them with recombinant interferon or with inhibitor that inhibit the interferon pathway. So with that, I will now introduce our first speaker with Jack Major, which is from originally is from Newcastle and is doing his PhD in the Francis Crick Institute, and is looking as you will see in his talk, he's studying the lung immunity in response to respiratory viral infections, and so how it's relevant for SARS-CoV-2 infections. And I will mute myself and let Jack start his presentation.
Great, thank you very much. Can you see this okay?
Yes.
Great. So yeah, thank you to Kwan and Nicholas for inviting me to speak. So as mentioned, I'm a final year graduate student at the Francis Crick Institute in Andreas Bach's lab, and here to to talk about today a recent publication will be published back to back with Achille and his team with Ivan Zanoni and other co-authors, where we both both sort of look at effects of interferons on lung repair. So, so following transmission from an infected individual, the respiratory viruses like influenza viruses and coronaviruses manifest in the epithelial cells lining the airways. And then following viral recognition, you get production of both type 1 interferons, so interferon alpha and beta subtypes, and type 3 interferons, including the lambda subtypes. And then these act as a frontline antiviral defense, inducing binding to their respective receptors, IFNA for type 1 interferons and the lambda receptor complex for type 3 interferons, inducing a downstream signaling cascade and the induction of transcription of interferon-stimulated genes. And this is a battery of hundreds of antiviral effector molecules that effectively function to induce an antiviral state in infected and bystander cells. And so they've long been studied as for their importance in controlling viral replication. So with respiratory diseases like influenza and COVID-19, you often, in cases of disease progression, the virus spreads to the lungs and the distal airways and can cause a significant immune cell infiltration. And in cases of moderate and severe diseases, you see increasing levels of epithelial damage and alveolar collapse and immune cell infiltration into lung tissues. So in the clinic, this presents as viral pneumonia. Patients develop a syndrome known as acute respiratory distress syndrome, or ARDS, and this can result in hypoxic respiratory failure, which is a leading cause of mortality in influenza and COVID-19 patients. Another complication is a secondary bacterial infection owing to a compromised lung epithelial barrier in that bacterial species disperse through the barrier and disperse into lung tissues, causing significant clinical complications. So it's important to note that the damage in your lung tissues occurs as both a cytopathic result of viral replication in host cells, but also as a result of an immune response generated by the host. And with diseases, respiratory diseases like influenza and COVID-19, host fitness depends on both the ability to control viral replication, but also to limit damage resulting from the immune response. So type 1 interferons, the receptor for type 1 interferons, IFNA, is also widely expressed in the immune system, and therefore interferon responses are also associated with inflammation, significant immune cell recruitment, and the production of pro-inflammatory cytokines and chemokines. And this can be associated with detrimental effects. So a previous study in our lab compared 129 mice to the Black 6 counterparts for their response to influenza virus infection. And it was shown that 129 mice had a significant increase in the level of type 1 interferons produced, as well as type 3 interferons. And they showed that the increased susceptibility of 129 mice compared to Black 6 mice was due to this exacerbated interferon response, as 129 mice deficient for the type 1 receptor IFNA had improved protection and reduced lung damage. And so this study showed basically that in situations where you have excessive interferon production, you can really exacerbate respiratory viral disease. So interferon lambda is more recently sort of studied cytokine, and in the last 10 years, it's starting to unravel its role in viral disease. So the receptor for interferon lambda is restricted mainly to epithelial barrier sites like the gut and lung, and also in neutrophils. And therefore, it's classically sort of associated with reduced pro-inflammatory responses and a far more targeted antiviral mechanism, therefore mainly inducing ISGs at the site of infection in epithelial cells. And this sort of improved, more targeted antiviral effect has again been shown in the study in our lab, where influenza-infected Black 6 mice were treated with either interferon alpha or interferon lambda after the infection, and both interferons were effective in reducing the viral load. However, only interferon lambda conferred protection, whereas interferon alpha drove a response similar to that in 129 mice with inflammation and lung damage, despite having increased antiviral effects. So this essentially shows that early interferon treatment has an effect in reducing viral replication. However, we still have to be careful in terms of how this affects your immune response, particularly with type 1 interferons. So critical for respiratory viral disease recovery is an efficient lung repair process. So to study this, so the importance of it is to sort of restore lung function and particularly barrier function, and maintain critical gas exchange function. So we study lung repair by flow cytometry. So we gate on epithelial cells in the lung by EPCAM staining and gating on CD45 negative cells. And then we segregate alveolar epithelial cells from airway epithelial cells based on MHC class 2 staining. And so what we look at in the sort of healthy, steady state naive mouse, the alveolar epithelium is relatively quiescent and turns over at a relatively low rate. However, following infection, it has a remarkable capacity to divide and repair, as we can see here at day 12 post-influenza infection, measured by Ki67 staining. And so when we look at this over time, we can see this sort of rapid onset of epithelial recovery, starting at around day 5 post-infection in our model and peaking in around 10 days post-infection. And so when we compare this to the morbidity of the mice, we see the recovery and weight gain in mice correlates really nicely with the onset of lung repair, indicating that lung repair is crucial for resolution of the infection and highlights its importance. So to compare the dynamics of lung repair with interferons, we measured viral influenza virus matrix gene expression in the whole lung and also interferons in the bronchoalveolar lavage fluid. And so what we saw was that the interferons peak at day 2 post-infection, correlating with the highest levels of viral load. And then with interferon alpha and beta, the type 1 subtypes, you see this decrease a little bit. And then they shut off around day 6 post-infection. The magnitude of the interferon lambda response is greater in concentration, but also in maximal peak production. And importantly, we also see it switched on for longer later during the course of infection. So why is this all sort of, and why we're interested in this in the context of lung repair? So in addition to the antiviral functions of interferons, they have a long history in anti-tumor therapeutics associated with the anti-proliferative effects. So this sort of makes sense in the context of the primordial interferon function, which is to stop virally infected cells dividing by shutting down the cell cycle or inducing apoptosis. So in the context of lung repair, we are really interested in how interferons may disrupt this lung repair if present during the onset of lung recovery. So as I mentioned before, interferon-associated pathology is usually studied in the context of type 1 interferons, in the context of immunomodulation and acute inflammation. However, this project, we also wanted to look at the effect of interferon lambda with the receptor being expressed on epithelial cells and to see how the 3 interferons may affect lung repair. Particularly looking in the context of endogenous interferons at this time window, whereas you see lung repair is initiated and you still have presence of interferons, particularly interferon lambda. So this is a question. And to start, we wanted to compare the 3 interferon subtypes at equivalent dosages. So whenever we treat in vitro or in vivo, we wanted to have comparable dosages after a short period of time. So we treated airway epithelial cell cultures for 4 hours with interferons, titrated, and then used this to generate a dose response curve and a conversion factor of equipotency. And then to start things off, we sort of kept it quite simple, and we just wanted to extend the interferon response further into the repair phase of infection. So we treated late during the course of infection at day 8 to 11 post-infection and measured proliferation by flow cytometry at day 12. And to study the effects of type 1 interferon specifically in the epithelium, as I mentioned earlier, the receptor IFNAR is widely expressed in the immune system. We used bone marrow chimeric mice where irradiated wild-type recipients were administered IFNAR knockout bone marrow cells, therefore restricting the type 1 receptor to the stromal compartment. And so in this model, when we treat with interferons, we see that both interferon alpha and beta significantly reduce the proliferation of the lung epithelium following influenza virus infection. And we see this is independent to changes in viral burden. And we think this is most likely because we treat quite late during the course of infection when viral control is less important. And we did a similar experiment for interferon lambda, and we see again that interferon lambda significantly reduces the proliferation of lung epithelial cells independent to changes in viral burden. And to study specific effects of interferon lambda treatment on epithelial cells, we depleted neutrophils who express the lambda receptor with an anti-Ly6G monoclonal antibody. And we show that when we deplete the neutrophils, lambda still reduces lung proliferation during influenza recovery. So that's sort of looking at exogenous application of interferons, but we next wanted to see in our model, do host-produced and endogenous interferons disrupt lung function? So we looked at day 8 post-infection, which is right in the middle of that window, crossing interferon signaling with lung repair. And when we compare IFNAR and lambda receptor knockout mice to wild type controls, we see an increase in proliferation for both, indicating that interferon signaling produced from an endogenous source disrupts lung proliferation. And we confirmed this was specific to the epithelium again in bone marrow chimeric mice when we saw a similar increase in proliferation. So actually one sort of question is that arose from this data was that in a system where one interferon system is lacking, why does the other one not control proliferation? So for instance, if in IFNAR knockouts, why do we not see interferon lambda controlling proliferation to the extent to similar levels of a wild type mouse? And so we wanted to look further into this. So we first of all checked the viral burden in the individual receptor knockout mice, and we saw very little difference across the time course, apart from a little increase at day 2 post-infection in IFNAR knockout mice. The similar control between the 3 genotypes is most likely due to interferon redundancy between type 1 and 3 interferon in controlling viral replication. So when we look at interferon production in these mice, what we noticed was quite interesting in that in the interferon lambda receptor knockout mice, they had pretty normal levels of interferon, both type 1 and type 3 interferons. But in the IFNAR knockout mice, we see almost absent levels of type 1 interferon, but importantly, we also saw significant reductions in interferon lambda, particularly at day 5 post-infection, which is important in the context of prolonged interferon responses disrupting lung repair. So we believe this is due to a steady state priming defect in the IFNAR knockout mice. So in a normal steady state wild type host, you have constitutive tonic interferon beta produced that consistently activates the IFNAR cascade, and this results in the basal sort of induction of signaling molecules important for interferon signaling, and also innate receptors. And these are really important for production of interferons upon infection of a host. So in the case of an IFNAR knockout, you lose this steady state priming, and therefore this results in impaired capacity for interferon production as seen in the IFNAR knockouts. So to circumvent this, we decided to treat with an anti-IFNAR antibody to block the signal from the point of infection at day 0. Therefore, you block the type 1 interferon signaling. However, you maintain steady state priming important for interferon production. So we effectively saw a nice blocking of the type 1 interferon signal. We measured ISG induction in CD45 cells depleted for neutrophils to look at specific effects of type 1 interferons. And we see this corresponds nicely with normal levels of interferon lambda production in the anti-IFNAR treated mice. So in a context where we have blocked type 1 interferon signaling, yet normal levels of interferon lambda, we no longer see the increase in proliferation indicating that the phenotype in IFNAR knockouts is a result of reduced interferon lambda signaling. And therefore, we think in our model, at least, interferon lambda is most effective in reducing lung repair, and at least at endogenous levels. So we wanted to characterize the phenotype further. So we performed an RNA sequencing analysis on lung tissue taken from wild type and lambda receptor knockout mice. What we saw, which confirmed our proliferation data, was an increase in pathways relating to proliferation and cell cycle in the lambda receptor knockout mice. And interestingly, we also saw increased pathway induction for genes relating to differentiation and multicellular genesis. And this is also a really core component of lung repair following damage, as not only do you need to proliferate, you need to differentiate, particularly in the airways, into multiciliated and secretory cells. And so I haven't shown you the data today, but in the interest of time, but we've also shown that lambda receptor knockout mice have improved epithelial cell differentiation, both in vitro and in vivo during influenza recovery. And so what does the improvement in proliferation and differentiation mean in these mice? So we measured damage levels when lambda signaling is absent, and we see that in the lambda receptor knockout mice, a reduction in the total and red blood cell number in the bronchoalveolar lavage fluid at day 8 post-infection, indicating that the improved repair phenotype in these mice correlates with improved disease outcome. And another way to sort of study these effects is to use a co-infection model. So as I mentioned earlier, in complication in humans is increased susceptibility to bacterial co-infection owing to a loss of epithelial barrier function. So you can test this in mice by administering bacteria. So here we use Streptococcus pneumonia. And when we do the co-infection model in chimeric mice to study specific effects in the epithelium, we see that lambda deficiency in the stromal compartment is enough to improve protection and also reduce dispersion of bacteria throughout lung and splenic tissues. So this sort of indicates that endogenous interferon lambda signaling not only promotes better repair, sort of dampens repair and disrupts epithelial recovery, but also enhances damage and susceptibility to bacterial infections later during the course of disease. So just to sort of sum up so far, we've shown that late treatment with interferon disrupts lung repair, and we think this has clinical significance, which I'll touch upon later, and which we'll discuss in the next 2 talks as well, mainly because COVID-19 patients also experience significant epithelial damage, and therefore we have to be careful when considering timings of treatments in these patients. We show that the effects are mediated directly in epithelial cells, and we also see the endogenous effect of interferon on lung repair. However, we believe that the effect in the IFNAR knockout mice is due to a reduction in interferon lambda levels, and therefore interferon lambda produced by the host is most effective in disrupting lung repair. And then we also see that this results in improved barrier function. So we next sort of wanted to look mechanistically, how do these interferons disrupt epithelial repair? So for this, we moved to an in vitro primary epithelial cell culture system. So we digest progenitor basal cells from a mouse trachea, and then we plate these onto a transwell where they proliferate, and then upon reaching confluence, we expose the cultures to an air-liquid interface, and this induces the program of further differentiation and proliferation. And then you've got a culture with a mix of cell types, including progenitor cells, secretory cells, as stained for by CCSP, and ciliated cells by beta tubulin staining. And so we like this as a model for repair in vitro because the proliferation and differentiation of progenitor cells re-encapsulates processes of repair following viral infection in vivo. So when we measured the equal interferon dosages after 5 days of treatment, we see that they effectively reduce the proliferation of both mouse and human primary airway cell cultures. And we see both in both mice and human cell cultures, the interferon beta and lambda have the most profound effects. And so we set up another RNA-seq experiment to try to understand mechanistically what's going on. So we treated growing cultures for 4 hours, but also a prolonged period of time for 2, 3, and 5 days to try and still see the effects on proliferation. So from our principal component analysis, we actually managed to get quite a lot of information from this alone, which I'll sort of explain through in bits. So the 4-hour treatment based on titrations for equal dosage clustered together, which nicely confirmed we did have similar levels of ISG induction after a short period of time. And this contributed to the variance on PC1 away from its respective mock. And we measured the genes contributing to this variance and quantified, looked at them by gene ontology analysis and confirmed that these genes are involved in pathways relating to interferon responses. So we assume that this variance is generated by ISG induction. And then when we looked at the variance on PC2, we found that pathways relating to this variance are involved in epithelial cell development and differentiation and proliferation. Which makes sense because these cultures, near the top right of the PCA plot, have been kept in culture for longer, and therefore we attribute this variance to that of proliferation and differentiation. So when we look at the prolonged period of treatment, we see 5 days of interferon, interferon lambda and beta significantly closer away from their respective mock, both on the PC1 and PC2, indicating a sustained interferon signal, but also a deviation away from the normal epithelial cell development. Whereas interferon alpha is a lot closer to the mock, indicating that are not as profound. And this correlates with our previous data we see in terms of cell growth experiments. And so when we look at the prolonged interferon-treated cultures, for pathway analysis, we see that interferon beta and lambda in particular induce pathways relating to cell death and proliferation. And then when we try to understand which transcriptional regulators mediate this, we looked at upstream transcriptional regulators by IPA analysis. And in addition to typical regulators of interferon function, so IRF and STAT proteins, we see regulators of cell cycle. So we were particularly interested in the tumor suppressor protein P53, owing to its identification in the gene set enrichment analysis in all 3 interferon-treated cultures. So to confirm the role of P53 in interferon-mediated anti-proliferative effects, we utilized P53 knockout cultures, and we showed that the effects on proliferation and cell number are abrogated when we in P53 knockout cultures. We also see that interferon-dependent induction of P53 related cell cycle genes, such as GAD45 and DUS5 proteins, no longer are induced in the P53 knockout cells, indicating that interferon induction of anti-proliferative molecules depends on P53. And so this is just a nice schematic from Matthew Hendricks and Ram Savan that was published a few days ago in Immunology, and sort of summarizes our work, as well as Akile and team, Ivan's team at Boston, basically showing that upon viral detection, you get production of type 1 and 3 interferons. And in vivo, at least, we see that the endogenous interferon lambda induces sort of P53 P21 mediated cell cycle inhibition, and therefore that disrupts proliferation and differentiation and affects barrier integrity, and this increases your susceptibility to bacterial co-infection. So I realize I haven't spoken too much about COVID, but I mean, I don't have these sort of, my data uses this influenza model, but we still think there's a relevance for COVID-19, particularly in the context of interferon therapeutics. So there's a lot of cohort data coming out at the moment, comparing sort of different patients in different categories of disease severity. And one thing that's starting to come out is a potentially presence of interferons longer term during the course of disease in severe patients. And this may interfere with sort of may increase lung pathology and interfere with the recovery of the lung tissues. So we think this is our data is relevant for this, because when we've shown in previous publications that when you treat early with type 1 and 3 interferons, this reduces viral replication. But importantly, in this study, we showed that interferon lambda in particular, if you treat with interferon lambda late during the course of infection, this exacerbates disease. And this makes sense as well, because if you later during the course of disease, viral burden is not as much of an issue, and it's more about controlling pro-inflammatory immune responses. And this was sort of corroborated nicely by a study that came out of Shenzhen in China, where they analyzed cohort sets from clinical trial data, and they showed that when they treated with interferon alpha as an aerosol nebulizer, early during the course of symptom onset, this improved patient outcome. However, late interferon treatment exacerbates disease. So while we need further work to sort of understand the immunopathology underlying COVID-19 disease, we believe that timing is the most is an important facet for interferon administration. So just to thank everyone. So particular thanks to my boss, Andreas, for the really great project and his support, and for the rest of the lab for all their support as well. Our collaborators in Aarhus and Freiburg University, Runa and Peter and Hans-Henrik, for their interferon, sort of giving us interferons to use in the lab, and also the training platforms at the Crick Institute, which have been a tremendous help. And thanks very much for listening.
Thanks a lot, Jack. It was fantastic. Quen, do we have time for question, or should we go right away?
Maybe one quick question. I don't think I see any in the chat, and you can ask question, or the speaker can have priority to ask questions.
Yeah, everybody, if you have questions, please just type it in the chat, and I will try to read them. Maybe I have one quick question. So, do you know a little bit more about this interferon P53 pathway? Like, is there any already data in the literature showing this pathway, and what does it involve? And did you try to give more, like, study more mechanistical, get more mechanistical insight on this pathway?
Yeah, so there's one paper published in Nature, I think it's in early 2003, from a group, and they basically showed that type 1 interferons directly induce P53, and I think they did a STAT1 chip on the P53 promoter, and this was mainly in the context of anti-proliferative responses. And in our system, we see the P53 pathway coming up after a prolonged interferon response, and this really corresponds with previous sort of more sort of biochemical and structural studies looking at how proliferative effective interferons are switched on. And it seems that they don't come on early, which makes sense in the host, in that you don't want to switch on these damaging genes early during the course of infection. You just want to stop the virus. But after prolonged signaling with interferons, then you start to see the anti-proliferative genes like P53 come on. So it seems that a prolonged interferon signal is needed to induce these pathways.
Yeah. So I'm very curious about that. Like, what would be mechanistically, what would change in the prolonged versus like a short induction of interferon? Is it some epigenetic change that would happen to switch the target, the gene's target?
Yeah, it's really an interesting point, and it hasn't really been sort of clarified properly. So a lot of the studies have so far looked at factors that contribute to the induction of these pathways. So they depend on things like receptor density, concentration. So a higher concentration of interferon is more effective in inducing these pathways, and really importantly, a prolonged interferon signal. So a 4-hour treatment, we didn't see any effect in our RNA sequencing in terms of switching on these pathways, but after a prolonged signal, that's when we saw them. And it may just be, it could be epigenetic factors regulating this, but it could be mediated by indirect mechanisms. We're not too sure.
Maybe we can take one question. One from so the first one is from Andy Min. Hello, Andy. So Jagger, nice talk and great work. Do you think the impact of interferon epithelial differentiation is primarily accounted for by impact on proliferation? Or do you think there are effect of interferon on both processes?
Yeah, so it's a good point. So we have data that sort of supports both sides. So we show that in the p53 knockout airway epithelial cell cultures, interferon lambda no longer reduces the differentiation of these cells, indicating that p53 disrupts the interferon induction of p53 disrupts the differentiation. And this makes sense because the multicellular genesis program is very coordinated in its cell cycle machinery. So inducing p53 and disrupting this would probably have an indirect effect on differentiation. But we also have data indicating that when you treat the cultures later during the course, when these cultures differentiate more than proliferate, we see an effect on differentiation independent of proliferation. So we've got a bit of conflicting data, but there could be multiple mechanisms going on. But yeah, it's a good question.
So there's another question. So from Eugenio Contreras Castillo, have you done an interferon-treated airway culture with poly-IC treatment to emulate viral infection? And will you expect any difference in the RNA-seq data in this experimental setup? So poly-IC versus interferon.
Yeah, so we haven't done that, and I think Akile sort of will speak about their model more, which uses the reductionist approach for viral recognition. But would I expect to see differences? I think it depends on the dynamics if how you used it. So poly-IC treatment, you'd maybe expect to see both type 1 and 3 interferons. So you'd maybe expect a amplified signal, but it depends on how long you treated it for, and yeah, the dynamics in which you treat. But no, it could be a good thing to try. But Akile's got some nice data to show you guys.
All right, so it's a perfect segue for me to introduce Akile.